Hi, my name is Guillermo Calero. I work at the University of Pittsburgh. I have my lab there. And today I'm going to talk to you about, I'm actually going to corroborate a lot of the things that you just heard. And I'm going to show you a couple of new ways to discover crystals and actually to find out possibly how well they will diffract. So the title of my talk is Applications of Transmission electron microscopy for Evaluation and Optimization of Crystal Growth. So I'm going to divide my talk in three parts. The first one is going to be use of transmission electron microscopy as a tool to discover nanocrystals. The second is going to be use of TEM as a tool to guide and improve crystallization. And third, I'm going to talk about new strategies for data collection that we have developed in the lab. So what are the challenges of modern day structural biology? Well, there's been over 100,000 deposits of structures on the PDB, but only 40,000 are unique, less than 4,000 are mammalian, and less than 11% coverage of the human genome, and mostly monomers. Right? So basically, we have barely touched the human proteome. So the most problematic targets, membrane proteins and multiprotein complexes. The problem with multiprotein complexes, for example, is that basically they require biochemical reconstitution, cannot be isolated by cells, so they pose an extra biochemical problem. So X-ray crystallography remains the most successful method to obtain structural information from biological targets. So there's been a lot of advances in protein chemistry that have allowed us to improve protein expression, also have allowed us to purify and solubilize more targets, and this actually has resulted in an increased number of samples available for crystallization. Similarly, advances in synchrotron science, such as the microfocus beams, the immune detectors available, and the software that actually make it possible to solve the structures. All of these have actually significantly improved crystal to structure timeframes. However, crystallization of protein remains the most significant bottleneck in structural determination by X-ray, and continues to have a significant negative impact in the field. As I said before, most of the P deposits are non-human proteins with a minimal number of multiprotein complexes, which are really hard to crystallize. For these methods, for these large multiprotein complexes, there are the current methods to improve crystallization, such as the protein thermostabilization or alanine scanning mutagenesis, etc. Generally don't work. However, as you know, and I'm going to barely talk about this, there's the development of serial femtosecond crystallography with the use of free electron lasers, which allow now to solve three-dimensional structures using nanocrystals. And so traditional electric crystallography uses one or several crystals. Of course, solving a structure using nanocrystals requires billions of crystals. And so this is an example of the samples that you need to use for BioXFEL crystallography. In this screen is shown lysozyme nanocrystals, which are 5 microns by 5 microns by 8 microns. And then you can actually see here the amount of material that you need to perform these experiments. So what you do is basically you take those nanocrystals, right? You put them on a gas-focused liquid jet, and then you basically you inject the crystals through this nozzle, right? And then you hit them with an X-ray, with a free electron laser, and then you get fraction. So, given that the problem is to crystallize protein, the interest in my lab was, can I discover nanocrystals of the hard proteins I'm working with? So basically, the question that we ask is, can we detect how can we detect nanocrystals of challenging proteins? So this work was done by Hilary Stevenson, which is a current BioXFEL fellow, and she did part of this work when she was a grad student, and now she's been in my lab for 1 year, and she has done tremendous advances in this. So basically what we did is we took nanocrystals of difficult targets, including soluble proteins, membrane proteins, such as these membrane receptors, and then multiprotein complexes, mostly RNA polymerase II complexes. And then here's an example of the gels for these proteins. These pink are the membrane proteins. These are multiprotein complexes, and this is just one of the soluble proteins that we use. And basically, we took Hampton research screen, and then first thing we did is we did BioXFEL microscopy. Right? So again, as Joe just told you, you get a spectrum of 9 or 10 possible conditions. I'm just showing you here 3, because this is just the most common that you see, as he said, clear drops, granular aggregates, and denatured protein. So we basically based our efforts on looking at granular aggregates. So again, when we set up Hampton Research Crystal Screen tool with one of the membrane proteins, right? As an example, we got 17%, 35% clear drops, 46% granular aggregates. We got a little bit of phase separation, and we got a little bit of the denatured protein. So this is pretty similar to what Joe was talking about. And this is just this is a membrane protein. So basically, we decided to focus on the granular aggregates to begin with. We actually did look at clear drops, but we couldn't find anything. But if I have time, I'll talk about it at the end. So basically, these are the examples of the granular aggregates that we saw. This is the one for the RNA pol2 transcriptional complex. Here are the soluble protein. This is a membrane protein. This is another membrane protein here. So first thing we try to do is how can we narrow down the number of aggregates? And because we already had we saw that we had a 40% aggregate, right? So the one way to narrow it down is to use UV fluorescent microscopy. So we basically work with Nagarajan, with Jansi, very closely, and he actually facilitated a lot of the equipment that we used to identify very small nanocrystals. So this is an example of one of the protein granular aggregates that we had, and you can see here basically in the right field there was no indication that we had here crystals, but when we look at it with a 40x objective of the UV fluorescent microscope, you can actually see that these samples were positive. Right? So basically, using UV fluorescence, we were able to narrow it down to a smaller number of aggregates that we were going to look at. So basically, then we decided to do electron microscopy because that's the natural way. If you're going to look at nanocrystals, why not look at them with the electron microscope? However, there were many technical difficulties to do this. People had already tried to do it, but basically Hilary had to work out a lot of the problems, and she actually was extremely successful. So the aims of visualizing nanocrystals with electron microscopy was just the first aim was to determine if our pre-screen samples were amorphous or were crystals, protein crystals. So again, we take these granular aggregates, and what we do is we take the granular aggregates and then we add molar liquor to the granular aggregate. Let's say we had 4 or 5 microliters, and then we put them on EM grids, dry the EM grids, and then add urine elastate. You're seeing here, and then just look at it on the very simple microscope T12. This is what we saw, right? So basically, for the samples that we were trying to look, we saw these nanocrystals. And here are some, you probably cannot see this for sure, but this is 0.2 microns, again 0.2. These are a little bit larger. This is 200, this is 0.2, 0.5. So you can see There are all kinds of sizes. Yeah, other sizes here. These are extremely small because this is actually 200 nanometers. So you can see it's probably 50 nanometers. And in some cases, we were lucky enough to see that we had a crystallize like this or like this. But in most cases, we didn't. So we knew for sure that these were protein crystals, but we didn't know these guys were protein or salt. Right? Here's an example of some amyloid proteins that we were also able to crystallize. So the first conclusion is, yes, we can identify nanocrystals from granular aggregates using electron microscopy. And yes, all the targets were positive. Okay. So the next question is, we needed to determine if the nanocrystals were protein or were salt. And to do this, we use a negative stain just to try to observe the crystallize. So why? The problem is that we encounter right away is that the nanocrystals were very thick. So how do you break them? So we basically tried to use the 4 millimeter Teflon ball from Hampton Research, and we actually were not able to break anything, of course, because they were quite small. So my lab is a yeast lab, and we actually break yeast using glass beads of much smaller diameters, 1 millimeter or 0.5 millimeter. So we decided to use those to see if we could break them. So we took the samples again, and then this is the 0.4 millimeter. These are the 4 millimeter, the 1 millimeter, and the 0.5 millimeter. We actually use 0.1 millimeter, but we completely annihilated the sample. So this one doesn't break, 0.1 millimeter annihilates the sample. So when we took these guys, right, we broke them, and we went down to the EM. Well, before that, I'm going to show you how it looks. This is before we break them. And basically, after we break them, you see a lot of crap here, right? But if we do the UV, you can see using the 40x objective, you can clearly see how homogeneous and nice are the sizes of these nanocrystals. So we actually were very excited. We took these guys to the EM, and then look at this. That's what we saw. So these were the first. These were basically the first really nice nanocrystals that we were able to see. And for example, here you can actually distinguish the fracture planes, right, in the crystal. And in this case, you can actually see these molecules of polymerase, which is a really large molecule. You can see them all the way here. Right? You can see them coming off. Now, before doing this, we wash the crystals thoroughly. So that means that this protein that you can see here, all these white particles, just came off from the crystals. And basically, when we took an FFT of the crystal lattice, we were able to identify bright spots. And I'm not sure how well you can see this because of the quality of the projector and this. But if you're interested, I can show you. These were actually 4 other bright spots. So that meant the lattice was quite ordered for this level of negative staining. Right. Here's another example. You can see in this case the small polymer, the large polymerase particles individually, right? And you can see also all sort of things. Here you see some probably aggregates of small, of little crystals, and then you have this crystal here. This is 50 nanometers. Right? So basically, for this guy, you're talking about very few particles in this lattice. And we were also able to see very large fragments like this, or very small fragments like this. This is 50 nanometers, so this is probably, I don't know, 150 by 100, right? So we were very excited about this. And in conclusion, what we saw is that fragmentation allows us to observe crystal and nanocrystal lattices for most samples, right? So basically, yes, we can see nanocrystals with TEM, and yes, we can identify if these crystals are protein or salt. Okay. So now, how do we take it from here? So The second part of my talk is going to be how to use TEM to guide and improve crystallization. So the first thing is, okay, we can now break every single nanocrystal using these protocols, and we can actually look at the lattices. So can we see differences in the FFTs or in the lattice among all these crystals? That would give us a hint of which crystals diffract better than others. Right? So basically, here's the answer. So this is some of our RNA polymerase transcriptional complex. You can see the crystal again here, and then before fragmentation and after fragmentation with FFT. It's a really nice slide. Okay? However, this is a membrane protein. This is a PTH receptor. Nanocrystals, right? Look at that. There's almost no diffraction. Okay? So these crystals, when we took them to CXI, they didn't affect. And here are other examples of several other proteins or protein complexes. You can see some of them are quite nice. This wasn't that good, but this is pretty nice. And this, of course, pretty nice. It actually diffracted quite well at the CXI 1.8 angstrom resolution. So here are more examples of lattices that we observed, and basically we also took to the synchrotron to see if we could correlate in some way the FFTs or the lattice with the diffraction of the synchrotron. So in this case, we have like a 4 or 5 angstrom diffraction of these crystals, and it was actually, you can see the FFTs clearly anisotropic, and the diffraction was anisotropic. Here's another example, 6 to 8 angstroms, another polymerase complex, and you can see pretty bad anisotropic diffraction. But here are other two that look quite good. 2 angstroms, nice FFT. 2 angstroms, also nice FFT, nice, very nice lights. Right? So yes, there appears to be a trend between how the lattice looks and the FFT and how the diffraction would react. So how else can we do with these fragmentation protocols and with this observation using them? So other things that we're able to see is, for example, the recognition of anisotropy. So all of these crystals that you see here had an isotropic diffraction. And when we took that to the synchrotron, we knew that they were actually anisotropic. So basically, if you start with something like this, I'm not sure how much are you going to be able to improve it. Here's another example of this crystal that you can see basically the lattice in this orientation. For this protein complex, you have a nice black spots here. The lattice looks quite nice in this orientation. And the diffraction is good. This is actually, this goes to 3, the angstroms here. But on the other orientations, you can see that you basically have no spots here. Right? So we saw this first, and we thought this might actually be anisotropic if we take it to the synchrotron. Then we did that, and then basically we saw that there was an isotropic. Right? Basically, this is how the lattice would look, and when we saw the structure, and we actually look, you can see that some orientation crystal pattern is quite good, but in other orientation you have these huge gaps that are just filled with solvent. The other thing that you were able to do with them is check for some things like non-uniform diffraction. And how this happened is quite curious, because we have these crystals. You can see them here, and this is the same crystal that was rastered at GMCA at the APS. And when we look at this crystal, you can see that most of you are familiar with this rastering process. The red spot indicates that you have a lot of diffraction spots. The blue spots indicate that you basically have very few or non-diffraction spots. So in this case, this crystal, which actually is outlined by this polygon, show large. I mean that in this area of the crystal, you had a good number of diffraction spots. However, in the other areas of the crystal, you didn't. So we basically, after we saw this, we took these crystals to some of these crystals back, and we basically look at them on the TEM. And Hillary spent quite a bit of time trying to find this, and he was actually lucky to see, for example, that in this area of the crystal, right, the lattice was quite well ordered, but in other areas, like, I mean, you had a good black spot, but in other areas like this one, right next to it, basically it was not ordered. And you can see it, you know, the particles are not really well aligned in this case. So that's another application of TEM, right? You can actually see overall how your lattice looks, and if there are some defects in some areas of your lattice. This is probably, you might actually be able to help these crystals by seeding or maybe by improving your protein purification protocol. Now, the other thing that you can see with them is evaluation of the solvent content. I mean, the main reason you can do this with negative stain time is because there's a high contrast between the solvent and the particles and the protein. So in this case, this is the lattice I showed you before. This lattice for RNA polymerase 2. So you can see how well the particles are, you know, protein particles are separated from each other. And actually, this correlates with a solvent content of 78 when you solve the structure. This is 78% solvent content. Here's another one actually that is basically almost the same particle size because it's a hexamer of a 60 kilodalton protein. So you have about 306 kilodaltons per liter inside 50. And you can see this is a, you can see the individual particles, but the lattice is much tighter. Here's another example of a 55% lattice. And you can see the difference clearly from this. And of course, when you go to 40% lattice, it's extremely clear that you can tell what your solvent content is by TEM. Why would this help you? Well, because I'll talk about it in a minute. Now, this is something that Joe talked about. This is another thing that you can identify with TEM. And basically, this is a protein that forms, that multinucleates, right? And it was really hard to improve. So we get all these bundles, or a little bit larger ones, but basically, you know, nothing you would do would help. And we took one of these bundles, we did it to the singleton, and you can see this is a 5 angstrom, right? And there's some spots here and some spots here that you probably cannot see. And of course, it's just not a single crystal, right? But this indicated that the lattice was probably, you know, well ordered. So we were curious what was happening here, why we couldn't improve it. So basically, we took this to TEM, and as you can see here, this is an example of that crystallization condition. So you can see all these aggregates, right? And then here outlined, you see a crystal, and this is actually shown here. So what you see basically is a nice crystal here, right, with a nice effectivity, and all of this extra kind of crystals starting to become crystals, right, or aggregates, right? So, I mean, we thought this was a very useful observation because from here, the problem was really that probably the protein was aggregating. So we needed to do something to the protein and basically use and apply protocols that would clearly slow nucleation, using probably detergents or additive screens or other things. But this was very reassuring for us. So the other thing that you can use this is for these experiments that we did at CXI in collaboration with Diana Cohen at Stanford. So basically we had the same protein, wild-type protein, and a mutant. But this mutant had absolutely nothing to do with crystal packing. It was inside the active site. And the purpose of this experiment is to see at room temperature how the enzyme would look versus the mutant, right? So the samples were absolutely the same. They crystallized the same way. And Aina took these samples to CXI. And then basically she got 4,000 hits in 250 seconds. So 16.4 hits per second, which is actually a pretty large number of hits for the mutant. However, the wild type, let me clarify, these 4,200 hits are at 2 angstrom resolution. Okay? For the wild type, only 670 hits at 2 angstrom resolution. with a much lower rate of hits per second. So they were really puzzled. They didn't understand what was going on. So I asked her to give me the samples, and we were going to look at them. So basically what we did, and I'm going to talk a little bit further, a little bit later about this. What we did is took the nanocrystals from each of the samples, and we put them on hemocytometers. And we count the nanocrystals using UV. So basically, you take your nanocrystals, put them on a surface of a hemocytometer, put a cover slip on top, and then take a UV picture with different magnifications. And basically, you can do dilutions of that. I'll talk about it in a little bit later. You can see that the end result of this experiment was that basically both samples had the same number of crystals per mil. This was 1.1 times 10 to the 8, and it was 2.4 times 10 to the 8. So indeed, this actually had a little bit more, but for this is not significant for the CXI setup. So we were wondering, okay, the number of crystals is the same, protein is the same, what's going on? So we then we used TEM to look at them, and then we saw that there were the Q lattice for some reason look at a much better order than the wild type lattice. This is the Q lattice. You can see the FFT, beautiful, and this is the lattice for wild type. You can see the FFT. It's actually not that good. And when we look carefully to all the wild type samples, we were able to see all these aggregates. Right? And you had two types of crystals in the wild type sample. Crystals that were actually that had a not well-ordered lattice, and crystals that had a well-ordered lattice. So the problem here was the prep was contaminated with another protein. So she said when the person that set up the trays, when she set them up, she didn't look at the sample. The sample had this contaminant, and basically the contaminant resulted in, basically polluting the crystals. And they had these two types of crystals in the same sample. So they did get some hits at 2 angstroms, which are here, but mostly were not good. And the other thing that happened to that sample is that the jet kept clogging. Right? So with this, we think that, and actually we're writing a manuscript about this, we think that TEM is actually a really good way to pre-characterize your sample before taking them to the CXI. Okay, so then what's the other application is we try to see if since we can actually look at the solvent content of the crystals. So for crystals that have solvent content, can we see if the crystals improve through dehydration? So the first example we took was something that was, I come from the Cornberg lab, so this was well known. What happened to solve the structure of RNA polymerase II is they basically did a dehydration protocol. So crystals before the dehydration protocol, they had problems. The diffraction was not very regular, and in some cases it was anisotropic. And at best, at 4 amps of platelet, more or less that's what, 4 or 5. They had very few crystals that diffracted well. So they applied a dehydration protocol, and they basically get the structure of polymerase. And we tried to see first if we were able to capture that using TEM. And so what we did is that we take crystals of delta 47 polymerase, and we look at them with TEM. You can see that it is clear. Right? Then we did apply a dehydration protocol, and basically we got an improvement clearly on the lattice. So we actually were able to detect changes in the lattice by dehydration, and then we decided to apply to another protein that the structure hasn't been solved, and we actually solved it in the lab. This is the DGTPase. And we had these crystals, and they were anisotropic. So we basically applied a dehydration protocol. Before we did the dehydration, 5 angstroms anisotropic. You can see here. Then we did the dehydration protocol. We got 2.7 angstroms. So we were able to do this because crystals. We were able to look at the solvent content of the crystals, and we thought maybe this is a good a good way to improve our diffraction. Of course, you can do this with crystals at the synchrotron, but if you have TEM and you can do these experiments, you can actually save a lot of time. Good. Now, here's another example of the use of TEM. So basically, we set up these crystals with TEM, very hard to crystallize. We set up some trays. We look at different conditions from the tray. Okay. So this condition has salt and PEG, and as you can see here, there's a bunch of aggregates all over the place, and the lattice has a really good quality. Another condition that had PEG but no salt actually shows better lattice, better order. So the conclusion is, can we look at, you probably can use TEM to look at your granular aggregates and decide which condition, if those granular aggregates are proteic, decide if you're actually going to, for which conditions are you going to go for. This, wait, I think I'm missing one. Sorry, I'm missing one slide here, but yeah, so now I've shown you exactly, that TEM is a good method to pre-characterize your sample, pre-characterize your crystals, and I'm going to now tell you some of the applications, right? So this is a collaboration we did with Alexandra Ross, and what she was trying to see, basically, she's going to give a talk, and we're going to hear from her this better. But basically, what she does is she created a device where she can separate crystals from different sizes. So what she did is that she took her, she takes samples and basically with crystals, and she's able to separate the small crystals from the large crystals or the larger crystals. So we wanted to collaborate, and she asked me if I could look at the samples with them. This is an example of photosystem 1. So this is the sample before sorting, and as you can see, you have large, very large aggregates of stuff. And then even here you have some probably bacteria, right? This is before the samples were injected in her device. After, and these are samples after sorting. So as you can see, she really achieved her goal to separate completely the large aggregates and the contaminants from the very nice size crystal. This is 500. This is 100 nanometers. So this crystal is probably, so this is 100, maybe 400 by 500, more or less. So these are some examples of the potential applications. Now I'm going to talk another application, which is seeding, and this is to grow large crystals. Okay. Ooh, sorry, I put this. This is the one that was missing. Sorry. So what you see here basically is another condition where we were able to identify granular aggregates, and this is what the type of granular aggregates that Joe was showing. And basically, we look at them with UV. This is a very hard protein to crystallize. We were never able to look at them in crystals. Then when we look at these granular aggregates, we are actually able to determine that they were actually protein crystals. And I'm not sure if you're going to be able to improve this, but at least you know that you have them and that you can actually try to improve them. So let me go back to seeding. Okay, so basically, this is what we did. We use these seeds that you see here, right? We have these seeds now. So how can we use this to really grow large crystals? So what we did is that we took these seeds, and as I said, we put them on chemocytometers, and the same way you count cells, if you take a UV picture of these seeds, which you can see are extremely homogeneous after fragmentation using the 0.5 millimeter bits. So basically, you take this. This is a 15x objective. Put your fragmented crystals, and then take pictures with different dilutions. And basically, you just proceed to count them as you would count cells. So here's an example again of what you would see. And this is an example of once you know how many seeds per microliter you have, you can basically now play with the number of seeds and see what are the results when you set up trays. So in this case, for example, we use 80 seeds per microliter, and we had actually no protein. We jumped into 160, and we actually started seeing larger crystals, 360, and then the crystals started to show, and they were not as large as the past. So basically, with this, you can do a very fine quantitative seeding. You can no longer have to use a hair from a cat or whatever, and then just magically try to see if you can reproduce. This is a nice way to look at the number of seeds that you have and to try to reproducibly grow every single time that you try crystals. So with these results, we basically were able to take, in this case, very small nanocrystals and grow large crystals, which diffracted up to 3.60. And we also took granular aggregates, and which actually that had nanocrystals, and we were able to grow larger crystals. And also, if you use seeding with a fixed number of seeds, by varying the protein to precipitant ratio, you can actually get these collections of crystals. Right? We call them catalogs. So in this case, what you see here is 2.5 to 1 protein to precipitant. You can see the large number of crystals. And then we keep going from 2.5 to 1.5, 1.5 to 1, 1 to 1, 3.5, 1. You can see the progression really nicely. So why is this useful? Because sometimes if you, for example, you have some protein that gives these crystals, but you want to try to get crystals for CXI, basically, that you would like to collect at room temperature, where you basically can apply this and get the smaller crystals. Okay, so the last part, I'm not sure how much more time I have.
You have 10 minutes.
Okay, perfect. So, basically, as I mentioned to you before, there are two ways to use, well, you will hear it much better from other people, but basically you have the injector. In the injector setup, you have to take nanocrystals and you have to put them through a jet and you have to diffract them, right? However, in my case, polymerase crystals have huge solvent content, 78% solvent content. So these are very mechanically fragile crystals, right? So when we put these crystals on the CXI setup, basically we were not able to get good diffraction. The best good diffraction we get was 4 angstroms. And when I came back from CXI, extremely disappointed, and Aina Cohen and I thought about the problem and she said, why don't you use an injector, right? Take an injector and see, do some experiments. And so basically we took the injector, she came, and then Hillary did the experiment. She took crystals nanocrystals before injection and after injection, and we did see differences, right? So you can see a little bit better FFTs here, the lattice is a little bit better. In this case, after injection, the crystals were basically in worse shape, and the FFTs are also in worse shape. Again, this is a very high protein, very high solvent content protein crystals, and also these are one of the first injectors, right? But for us, that was a problem. Second problem is that for me to grow this number of crystals, it requires, growing, I don't know how many hundreds of liters of polymerase. So for me, that's not really a good scenario. Of course, things have gotten better now, and injectors use much lower quantities. Things have improved. But for me at this time, I wanted to be able to shoot my nanocrystals, the nanocrystals that I had discovered. So the question was, how can I collect data from crystals that were mechanically sensitive and also how can I collect data from nanocrystals that were actually too small for the the quantities were basically not enough, or not the quantities needed for the jet setup. So basically, we had to develop some way. So what I did is, in my lab, what we did is we took nanocrystals from these very nice multiprotein complexes, polymers with TVP, TF2B, and TFG2, right? And here's the sample, very nice, whatever. But how do I fish these nanocrystals? So the first thing I did is I took a cryo-EM grid, right? And I just epoxied the cryo-EM grid to a TEM setup. And that's the way it looks. You can see the epoxy here, right? This is a copper grid. And that's the way it would look with bright field microscopy, right? So you can see some crystals here, right? But then when you use the UV, look at that, right? So I was able to identify less than 20 micron nanocrystals on an EM grid. And basically, if you overlay these two pictures, you can actually get the layout of each one of the nanocrystals using very simple software developed at the NIH, ImageJ. Right? So with ImageJ, we were able to get all these and all the positions. So then we wanted to see if these crystals would just diffract. Just proof of concept, we took them to SSRL, sent them to INA, and talked to with user 10 by 10 micron beam, and then we shoot these crystals that were smaller than 20 microns with 40-second exposure, no attenuation, and we actually see the fraction to 6.5. And then we actually get the 80 seconds, I didn't put it here, I'm sorry, but in 80 seconds, I fry the crystal, of course, so I get better fraction, 5.0. So yes, those crystals would probably diffract. The proof of concept was there. We were actually able to get the copper grids and then shoot the nanocrystals. However, as you can see here in this picture, how many do you see actually not lying on the copper? Right, very little. So it was impossible to basically have this large number of nanocrystals shot by the X-ray, because a lot of them had the copper background. So what we needed to do is basically design a new set of holders for these nanocrystals. And then what we wanted to do is identify these nanocrystals using UV, because as you saw, it was very clear, the positions of the crystal were very clear when you do UV. When you did bright field microscopy, we were not able to see them at all. And the third was, can we determine the spatial coordinates of the nanocrystals on the grid with respect to some reference points? And basically, you probably can guess the answer is all of this. So basically, we started doing this when the collaboration mainly with Arna Cohen, and now we're starting a collaboration with Pegorata and APS to do this. So what we did is we made these grids, which are made of mylar. Actually, this is the latest generation. The first ones I printed them. And you can see here the nitrogen loop, and this is our grid. And this is a crystal drop at scale. So this is a 1 microliter crystal drop. So what you basically do is you take 6 or 7 microliters from your mother liquor and you add it on top of this crystal drop, and then you get your loop, and then you get everything. In 2 seconds, you get the whole thing. And then what we do is that we have this UV setup, and again, we're still working on it. So this, you can see this is a UV microscope, and this is the wand, and you can see the crystal here, right? UV. And then we take a picture, and that's what we get. So
frozen, or is that still?
No, that's not frozen. That's not frozen. Yeah, but it's not frozen yet. Sorry, let me show you. So this is not frozen, just like that, right? So you take a picture, and that's exactly what you do. You freeze it. So these are some examples of crystals that were frozen on these loops. Again, this is the first generation that we had, and we actually printed this but the results were not very good because you can see how the loops have some defects, right? And it was really hard to be able to build some fiducials on this. But you can see that they actually serve their purpose, which was to be able to identify nanocrystals. And I'm not sure if you guys all the way there can see these little guys, but they're actually really small. These are 5 microns. These are basically 30 microns, and these are 150 microns. So basically, you can do any size you want, and you get hundreds or hundreds of crystals here, dozens here, and a few dozens here. And so basically, this is again with SLA. So this is an example of the procedure we did. We take the crystals, we put them on holders, we expose for 0.5 seconds UV, and then basically we freeze them. Once we freeze them, in the beginning, again, and this was everything in collaboration with Ina Conn, because she was the one that we wanted to do this automated. So we wanted to have choose some fiducials so that they could basically get the position of the grids on the so for the correction. So basically, we got the crystals, and this is the bright field image. This is the fiducial points that we chose, which basically are not very accurate, but you can see them there. And then these are the profiles for each one of the crystals generated with image J. And these basically are each crystal number, the area of the crystal, and then the positions with respect to these fiducials. To the 0, 0, which is fiducial 1 is 0, 0. So each one of these crystals can be positioned with respect to this. So then we actually took this setup and we tested it. I now already developed the software interface. So we basically took these samples for, this is XPP setup. We took these samples and then we gave them a list of the coordinates. They actually read those coordinates and they immediately generated these green rectangles around each one of the crystals. And this is just a very short movie. I'm not sure if I can get it activated. So you can see there's no step. Maybe there's one that's activated. Not sure. Yeah, so let me see if it's moving or not. So yeah, so you can see fluorimeter moving, shoots the first one, right? And then goes to the next, and goes to the next button. So basically you can get 100 crystals, 200, 300, whatever number, and you just hit it once you get the flash. Now with this XPP setup, the hole that you generate is about 30 microns. So if the crystal is 50 microns, you can shoot it once, but if the crystals are larger, what do you do? So I'm going to show you what do we do. Oops. Well, that's another movie, but I can show it to you later. These are the results of those shots. You can see this was polymerase, and you basically can see spots of 2.9, which is actually quite good for one of our complexes. And this is a shot on the Mylar loop, which is the third generation of loop that we use are actually made out of Mylar, less super. And you can see the background of the Mylar. And so basically what we did now, this is the latest generation, but this is for larger crystals. So you can see the brighter image. We take the UV, see how nice it looks. Then if you overlay the grid with the crystals, you can see that these are the ones that you would see some background because they are on top of the Mylar. But for all of these guys, you won't see anything because they are just floating on the grid. Right? So these are much easier to generate our anchor points, and we also break the symmetry by adding this arrow, so pointing to the first fiducial point. And then what we did is, okay, if we have a 200 micron or 250 micron crystal, why waste it with one single shot? So basically generated masks, and we're able to now generate, these are the list of points here. And these are separated by 30 microns. So basically what you do is you get a list like this now, and then you can just go and shoot automatically. You don't have to. If you see the XPP setup is really hard because if you don't have this type of thing, you will have to look into usually a 0.3 or 0.5 mitogen loop, and you have to spend 20 minutes there trying to find the crystals. In this method, you can get them right away, no matter what size, and you can shoot them automatically. And so basically, I think I don't have time on this with this. And just acknowledgements, of course, BioXFEL for providing funding for Hillary, which she did a tremendous job doing EM. NIH that has funded my other research, and the University of Pittsburgh, of course, for housing me. And these are grad students in my lab, other grad students, Christopher Vance and Lynn, which work on X-ray, and Du and Shelley and Jim, which also work on X-ray projects. And there's some other collaborators from Pittsburgh and of course, from Stanford, Anna Cohen. And well, thank you very much for inviting me.